Thanks for the kind introduction. And I want to first express appreciation to the organizers of the meeting for inviting me to give a talk about the research work that is going on in our lab. The first part of my talk will give an overview of our research directions before we enter into microbiome, which is the topic of the day. You can see here is a photo panel of individuals of diverse ethnic background from different countries of the world. Our lab has long been interested in studying differences and similarities in people of diverse ethnic background. And some of our key questions here have been to look at how ethnically diverse humans differ in regard to genomic and phenotypic variation. And also how is evolutionary processes that drive and maintain this variation. The complex traits also involve the interaction of different genes, genes also with proteins, and the contribution of the environment. And the lab has also long been interested in seeing how this interaction brings about variability in these phenotypes. And most of our focuses see the, as has been described in the literature and also by our group, most of diversity is seen in Africa. As you see here, in these individuals are different from different parts of Africa. So most of our focus has been in Africa. And talking about diversity in Africa, to give you an idea of how Africa diverses, which is in size, Africa is the second largest continent in size and population after Asia. And also, if you look at the map of Africa, basically it can contain countries like the United States, China, India. Most of the countries of Western Europe can all fit in that, just to give a picture of how big Africa can be. And climate-wise, we have a lot of variation from the desert towards the north, and then you go to savanna and forest. You have all this diversity. And looking at the lower panel on this slide, the lower panel on the left shows genetic ancestries, which our lab has been one of the first to show how diverse Africa is. And here you see the different populations that were studied. You see the color panel shown there, which shows the contribution of different ancestries in each population. And you can see the different colors showing different ancestries, meaning the contribution of different ancestries in particular populations that were studied. And this was also picked up in the 1,000 Genomes project, the figure on the left, which shows on the y-axis the variant size per genome. And looking at the populations that the 1,000 genomes looked at, Africa, which is highlighted on that figure on the right, the African populations had the highest number of variant size per genome. Despite this great variability, diversity that has been seen genetically in African populations continue to receive less attention in genomics research. This is shown here in this figure, a study of 2016, where only about 3% of African samples were involved in genome-wide association studies. So Africa is still so much underrepresented in genomic research, despite this big genetic diversity that we know in African populations. What have our group specifically been doing? So in African population, I've looked at population structure, which gives an idea also about the history of the population, the different ancestries. And very elaborate also, they've looked at genetic variation related to skin pigmentation in African populations, and also adaptation to diet, which we publish on lactose tolerance, bitter taste perception, and relation to amylase. And recently we've gone to Gut Microbiome, which the first paper from the group just appeared online this week, and which I'm going to show you some of this data in this talk. And then we will also be moving on to study diabetes risk in populations of different ethnic backgrounds in the African continent. The approach of the lab looking at these complex traits is applying the omics sciences together with the contribution of the environment, as shown here in this figure. And like today, we were looking at the microbiome, which is part of the bigger picture that you see here. I will say different aspects of the study. In the lab, different people look at metabolomics, different genomics, proteomics, epigenomics, and also with collaboration with African collaboration and outside African collaboration here in the US and different parts of the world. And talking about microbiome, maybe a first definition of what we mean when we talk about human microbiome, we are referring to the genomes of all microorganisms, including bacteria, viruses, fungi, and protozoa that live inside or on the body of humans. In this figure here, which is a study published in 2016, and they clearly demonstrated that microbiome varies between indigenous or non-Western society when they compared it to the urban society. The populations that were studied in this, that were looked at in this study included the Ba'aka pygmies, which are rainforest hunter-gatherers, and they compared with Bantu and a population from the United States, a Western population. And what did they see? They could see a decline in gut microbiome diversity, higher diversity in indigenous population compared with low microbiome diversity in a Western society. And also they see differences even at the level of the taxa in the non-indigenous population, where you have Provetella taxa bacteria as the main bacteria and Bacteroides was highly reduced. And this was the reverse in the Western samples. So we're talking about here showing the gut and why the study of gut microbiome. Here, different locations in the gut, from the duodenum to the jejunum and the colon, you see different types of bacteria and also different abundances of bacteria. It's also with the colon having the highest type and abundance of bacteria, even though one can also argue that other parts of the gut might be less accessible to study. But generally, if you look at the functions of this bacteria also, you could still as well expect that the colon could have the higher abundance. And why do we have this motivation for studying gut microbiome in Africa, looking at it? So you might be interested in looking at bacteria diversity between the different indigenous populations to compare how gut bacteria diversity varies, and also, as been shown in other populations, what is the contribution of different diverse ancestries that we have, dietary habit and geographical location on microbiome composition, on gut microbiome composition. And looking also previous studies, I just showed the Ba'aka pygmies and some agriculturalists, but populations like the pastoralists have not really been looked into, or there's limited information about that in the literature about the gut microbiome for the pastoralists. And we also are interested in looking at whether the Western society that has more studied the microbiome has been more intensively studied, what compares with what we will see in the African continent. And finally, gut microbiome has been associated with disease states, and you might be interested to look at what occurs in African populations. And talking about gut microbiome also, you want to, what are the factors that affect the composition of gut microbiome? And here are this describing this study. Medications affect, of course, the composition of gut microbiome. You can think about antibiotics, that people on antibiotics or would affect their gut microbiome. Also, geographical location. Our lifestyle stages, children compared with adults and the elderly, we have variation. And the birthing process has also been shown to affect the microbiome of the infant and the feeding, and also disease. And talking about the contribution example here of how diet can affect our gut microbiome. So when you vary your diet from the high-fat diet to someone on a vegetarian diet or a more calorie-restricted diet, you can see the different types of bacteria here. The abundances vary when you are on different type of diet. Most of them shown here, just a selection to demonstrate, show an increased abundance in high-fat diet, but a decreased abundance in vegetarian diet or calorie-restricted diet. When we involve in microbiome studies, this slide shows about the ethical or legal issues that come into that you should consider when looking at microbiome. First of all, you have the host DNA in a microbiome sample. So this means you have to protect the identity of the individual. You have the DNA can be found in the microbiome sample. And secondly, what has been seen is that most people just sweep microbiome together with other studies, and they have a general informed consent that it's better to have a specific informed consent that covers all aspects of microbiome. And now with the coming of most microbiome biobanks, there is an ethical question of how whether the results should be returned to participants, because it could be incidental finding or result of medical importance. And another question also comes in on what structures are there to properly deliver these results to the individuals. And finally, we should also protect the environment, because when we work on microbiome samples, they contain microorganisms, and we might contaminate the environment with this sample, so we have to be careful. Now coming to what we actually do with our microbiome studies, what is the aim? Which is mainly we are interested to characterize gut microbial composition between ethnically diverse African groups living traditional subsistence lifestyles. And we compare this with an urban population from the United States. And what we're looking in actually is to determine if any taxa differ significantly, and if and composition, and if there are differences, whether these differences are attributable to diet, to geography, or to ancestry. Starting from here in 2011, we started sampling Tanzania in 2011. And as you can see, the populations that were sampled from Tanzania, and you have populations like the Maasai that practice pastoralism, and then you also have other populations like the Hadza who practice hunter-gathering, and then you have other populations that are agriculturalists combined with some limited pastoralism. And also similarly, populations from Botswana. We have samples also from Botswana, shown here from the San, the Herero, and the Bantu populations in Botswana. And most recently in 2015, we also moved to Cameroon, which probably has which we have had the highest number of collection. Most of the data that we showed today would be our first analysis, which will be focused on the Tanzania and Botswana samples compared with the US samples from Philadelphia, as mentioned. This figure shows some pictures from the field, from different field sites. So you can see settings on the field. and most of the people that are sometimes involved. Here, these pictures are from field site in Tanzania, Botswana, and Cameroon. And talking about microbiome, here showed how the samples are processed and transported, and just to probably emphasize that for good data, there should be emphasis on sample collection, sample processing, and transportation, and this will pay a lot of attention to it. Here, with my colleague on the field in the Baka village in Cameroon, processing microbiome samples. But first, we ask the individuals, the participants, to avoid fresh samples in a sterile container. And you must emphasize these are fresh samples, and we provide a container, and they have to give us. We don't take samples that are collected at home. The samples are voided immediately. And since we work in areas where enteric parasites are also predominant, so on the field, as a return of results to the participants, we screen for enteric parasite. That's why you see here microscope. We screen for enteric parasites, and the results are returned. And sometimes we have medical physician who we give us back to the participants who have been treated for this enteric parasite. But the aliquots for microbiome studies on the field conditions, so we use liquid nitrogen to store the samples. So aliquots for microbiome studies are stored in liquid nitrogen. And when we move back to the urban centers, the big cities where a -80 freezer is available in a collaborator lab, then the samples are transferred to -80 before shipped by world courier whenever it's possible to the Tishkop lab in the US and stored again at -80 until analyzed. What approaches are there for microbiome studies? Here, the upper panel, the 16S based approach is a marker-based approach, and in this you have amplification of section of the 16S RNA gene. That is the amplification of a region of this gene, and the sequences are grouped into different operational taxonomic units based on their similarity. And with search in the database, you will identify which bacteria, which sequences are coming from what bacteria, and then we can look at the abundances of these OTUs or look at phylogenetic. The second approach, which is actually, I would say, a better approach, the shotgun metagenomic approach, which looks at the total gene content in the bacteria and gives an additional advantage that you can look at the function of the different microbiome composition of the different bacteria that are present in a sample. We use the 16-base marker approach for the analysis of our samples. And here you look at the different methods that are used for extraction of these samples of DNA from the fecal samples, and the V1, V2 region is the region that is targeted in the 16S, and usually we have about 65,000, we go up to about 65,000 read depth per individual, and the region that is amplified is just over 300 base pairs. For data analysis, you have the CHIME, which is the quantitative insights in microbiome, microbial ecology, and this program you use to do analysis, look at the diversity in bacteria composition, both within individual and between individual diversity. You also look at principal coordinate analysis, and also the ANCOM program, which is analysis of composition of mycobacteria. We will see later how these methods are applied with the data I'm going to show you later. And of course, the R and the Python statistic programs are also highly used for analysis. Here on this slide, like I said, the data I'm going to show today is focused on samples from Tanzania and Botswana. and you compare with samples from the USA. And here this shows the different subsistence groups and how many populations were included. Most of this sample collection in the field was coordinated by Alessia Rancharo, who has done most of this field work. And back in the US with the analysis, the studies that I'm going to show have been led by Matt Hansen and Megan Roble. Matt Hansen's research associate in the lab. Megan Roble is a PhD candidate who's working actively on this microbiome studies. And our lab has worked in collaboration with the Bushman lab still at the University of Pennsylvania. So the data that I'm going to show is, sorry, Rolanda. So is everything okay there? I get some noise.
Yes, sorry, now I've unmuted.
Okay, thank you. So here the data I'm going to show is from this paper that has just appeared online this week, and which on genome biology with free access, which people are free to can access the paper. Here on this, starting with the results part of my talk, on the y-axis here shows the proportion of each bacteria in the total OTU counts and the populations that were studied on the x-axis. And two common bacteria, the Bacteroides, the Taxa, the Bacteroides taxa is most common, as shown here in the extreme right, in the US population, and this was less common in the African populations on the left. Meanwhile, the Prevotella is more an African bacteria, can also distinguish the African samples from the US samples. Also observed that in the African samples, the Prevotella varies within the different populations. So this is as expected, and this is something we confirm. One observation we saw also was that most of the unassigned taxa, the ash color in this picture was higher, highly represented in Africa compared with the US. Again, here with another kind of analysis, the ANCOM analysis, as you see in the whisker plots, the Prevotella bacteria and the Bacteroides, you see great differences when you compare between group comparison, rural population, the African samples compared with samples from the US, the Philadelphia samples. Surprisingly, we also saw that the differences in these two bacteria were also significant when you compare Tanzanian samples with Botswana samples. This we were not expecting. We thought these are samples from rural Africa. We thought they were going to be comparable, but that turned out not to be the case. And also other bacteria like Suterella and Treponema also showed significant differences between Tanzania and Botswana. And when we look at also, there was significant variation in a number of bacteria taxa across all the populations that we look at here, including the Bacteroides. Maybe I should highlight that on the y-axis we have the relative abundance in log scale, and the different populations that are studied on the x-axis. So these are bacteria that varied across population, across the different populations that were studied significantly. And I go to the lower panel, I would like to highlight the Hadza, which is a hunter-gatherer population from a hunter-gatherer population which showed a very high abundance of Treponema bacteria, and this bacteria has also been seen in other studies in hunter-gatherer populations elsewhere. And we also, when we group here on this panel, I'm showing bacteria that was significantly different by subsistence. And here again, Treponema bacteria I must highlight was more significantly of higher abundance in hunter-gatherer population. And to also probably mention that this bacteria has been associated with diets rich in fiber. Also significant between the subsistence group that we have comparing hunter-gatherer, pastoralists, and agro-pastoralists, the Ruminococcus was significantly different, but this is relatively a very common bacteria. The P75A5 bacteria showed are significant in our study population, and this is a bacteria that has been found in the literature. You see has been found in Bangladeshi children, and mostly found in ruminants and young cows. And we think that the higher abundance of this bacteria in pastoralists could be because of the association with cattle. In this figure showing the principal coordinate analysis, in which this shows the distance in the phylogenetic distances between the samples that we analyze, and here samples of a greater similarity clustered together. On the left would be mostly samples from Africa, and then on the right here you see the clustering of samples from the US. And again, surprisingly, we see a clustering of samples from the US, more with samples from Tanzania, from Botswana than samples from Tanzania. It was a surprise for us to see that samples from Botswana are clustering with samples from the US, even more than clustering with samples from Tanzania, and I'll come back to this later. But here to indicate on the x-axis with up to about 46% of variation is on the principal component 1 is explained as seen here on the s-axis, and then principal component 2 is able to explain about 12% of variation. And what we see also that the variation in phylogenetic distances between samples correlates with the common bacteria. Here the Ruminococcus and Bacteroides here clustering on the right, which are more common in Western samples, and the Provitella on the left, which is more common in African samples. And we can see the two panels agree. Another kind of analysis we look at was also looking at what is called non-alpha diversity, but here is the within individual, which is shows the within individual diversity bacterial composition. And here to the left of this, the Vowlin plots, purple shows African samples, and you have the US samples. And the diversity, this shows that diversity in African samples was much higher than what you see in the US samples. We are talking about within individual diversity. And looking at the African samples, the Tanzanian samples showed higher within individual diversity, because bacterial composition had a higher diversity in Tanzania compared to Botswana. And when you look at the populations individually, you also see significant variation across the population that was studied, with the Sandawe having the higher within individual diversity. Just maybe to indicate that Sandawe is a former hunter-gatherer population. And we guessed that maybe this is because there were former hunter-gatherers that have now settled down to practice a more agricultural life, and maybe this transition is accounting for this difference in the microbiome composition, the transition from hunter-gathering to another lifestyle. Another parameter that we also look at is inter-individual diversity. So we look at how dissimilar bacteria from two individuals from the same population vary. And in this, the US samples comparing with the African samples, the inter-individual variability or diversity was higher in the US samples compared with the African samples. And when you compare Tanzania and Botswana, Botswana samples were more like in the direction of the US samples, where there was a higher dissimilarity or diversity, beta diversity between individuals compared to Tanzania. and also like it observed in panel A for alpha diversity or within individual diversity, inter-individual diversity also varied across the different populations. In panel B, what we used in the metric was present absence of bacteria, but this is also confirmed even when you look at the abundance of the bacteria. Here on this figure, it shows on the y-axis phylogenetic distances or inter-individual variability. On the x-axis, you have within individual variability. For some unknown reason, there is a correlation between inter-individual variability, as shown here, negative correlation with within individual variability. On the other end of the spectrum, you have the US samples, and on the Sandawe and the Tanzanian samples cluster at the other extreme, and in between you have the Botswanan samples. This observation was has also been seen in some other populations that have been studied, but it's not clear why you have this clustering. We were surprised in this. Also, what we expected was that if diet was actually associated with gut microbiome, that is what we expected. We have a broader diet in the European, in the US samples, then you would have expected a clustering by subsistence where the hunter-gatherers would cluster together and the pastoralists would cluster together. But this is not what we see here. So this is something still intriguing about why you have this kind of an observation. We move further also to look at pairwise comparison, the F statistics, which compares inter-individual or different groups, samples from different populations, and look at phylogenetic distances. Comparing, we find that the highest dissimilarity was between samples from Tanzania, panel A, the first red bar on panel A shows that the highest differences of dissimilarity was between samples from Tanzania compared to the US. And next to this is samples when you compare Botswana to Tanzania and samples from Botswana and the US. What you see, which was also surprising to us, is that Botswana, as already highlighted in other analyses, Botswanan samples tend to be more similar to US samples than they were to African Tanzanian samples. In this, you can ask what differences could be. This also is not known what the differences could be. But when you look at economically, Botswana has a higher GDP and probably access to healthcare and other differences, maybe pathogen screening, the different kind of pathogens in the different populations might, to some extent, maybe explain why you see these differences. And we also see here differences according to subsistence, comparing samples from the US with hunter-gatherers, agro-pastoralists or pastoralist population. But also, like we said, we were expecting that when you look at the different subsistence groups, when you compare in the African population, agro-pastoralists compared with hunter-gatherers, that we were going to see a big difference if diet had a big contribution in microbiome composition, in gut microbiome composition. Here we didn't see that strong contribution. We see that, as shown in panel A, the geography was stronger associated with gut microbiome composition, or the similarity between samples from different sites more than differences between subsistence. However, we must also indicate that we didn't look at we didn't collect dietary data, and probably maybe you look at that and particular substrate, you may see the differences. We also somehow see that there were differences in gut microbiome between females and males from Hadza and Masai, but these associated bars, which shows the level of significance. The red bars show the level of significance. This was quite limited. So what can we conclude from this pairwise analysis here? We saw that the bacteria composition between Botswana and Tanzania are more different from each other than between any two subsistence groups. We see, though limited, sexual differences were higher in the Hadza and the Maasai. There were also some other analyses that were done that I didn't show the data here, but we saw that gut bacteria, the alpha diversity within individual diversity in bacteria composition was higher in people with low BMI. And also that bacteria similarity was correlated with geographical proximity and relatedness. So unrelated between individuals. Just to summarize what we have gone through and what the results of this study show, first is that within individual bacterial diversity was higher in Tanzanians compared to Botswana. And this varied also across all populations and indicate that Sandawe had the highest within host diversity across all the populations, the individual populations that were studied. The similarity of bacterial composition between individuals increased with geographical proximity and relatedness between individuals. Here I must highlight that geographical proximity had a higher effect on bacterial composition, even when we look at presence, absence of bacteria and bacterial abundance. However, relatedness was a bit limited, and this was restricted to when we look at presence, absence, and in abundance, this was not significant. Subsistence type, as just mentioned, had a limited effect on bacterial composition compared to geography. And we observed that the trend between diversity and dissimilarity followed what has been observed in industrial populations or Western populations as expected. Like highlighted, the Botswanan samples were more similar to the US samples than the Tanzanian samples, and this is probably something of interest to look at what exposures in Tanzania, in Botswana, that distinguish Botswana from Tanzania, and that are more closer to the US population. And we think that shotgun metagenomic studies on some of these samples that we are doing might shed more light on some of these differences. What are the future directions? First, we've shown quite a limited number of samples in our studies, which sometimes could account for some of the non-significances or the differences that we see. But we have a higher sample size from Cameroon, and this from the different populations also, including hunter-gatherers, pastoralists, and agro-pastoralist populations. And this will probably shed more light on some of these findings that I've shown you today. A subset of these samples, we are doing metagenomic shotgun analysis, so we can look into the different bacteria composition together with function. It is not known what drives the trend towards westernization, and we're looking at pathogen screening, and I will also include that we are looking at also cytokine screening in these individuals, and we think this might shed more light on this observation. And also we are interested in looking at association of gut microbiome composition with fasting glucose, with lactose phenotype, and also with BMI. And this we're going to analyze in the big data set from Cameroon, which we have some diabetic patients, and we have data on glucose and lactose phenotypic and phenotypic data. And finally, it would be interesting, we just look at a cross-sectional approach, but it would be interesting to look at longitudinal studies in combination with collecting dietary information. And that comes to the end, and this is the most important slide in which I want to thank the members of the lab, the Tishkov lab in the US that did most of the analysis. Megan Rubel and Mark Hansen are those who did most of the analysis and shared first author on the paper that I've shown you, and other collaborators in the Tishkov lab. The Bushman lab also, where most of the analysis was done, and also at the University of Pennsylvania, and collaborators from University of Yaoundé 1 for collection of samples from Cameroon, University of Botswana, and the Mumbili Mohas University in Tanzania, collaboration also from the National Cancer Institute, and appreciate all the sources of funding from NIH, and other sources of funding that we have for these studies, and the different governmental institutions and ethics committees from the different parts, different countries where the studies, the samples were collected. And most importantly, the study participants, otherwise the studies would not be possible. Great thanks to them, and thank you so much for your kind attention. Thank you.
Okay, thanks, Eric. All right, does anybody have any questions? You can either,
Hi, Eric.
Yeah, go ahead.
Can you guys hear me? This is Matt Bramble in Washington, D.C. Our group has recently done about 300 metagenomic sequencing on stool from individuals in the DR Congo.
Right.
And I don't know in your samples if you're seeing any of this. We have levels of actinobacteria that are just outside of anything that I've read thus far. The samples are pretty much, as a whole, just actinobacteria and Firmicutes, and nearly no Bacteroides. If you look at a phylum level.
Right.
Have you seen any? Have you seen this in some of your African populations, or it's I'd say 40% in some of these samples are actinobacteria.
Wow.
And yeah, I don't know if this could be a sampling error, but I mean we've seen it consistent, and I don't think it's that.
This is interesting to what you mentioned. In the Cameroonian samples, yes, we are looking at Archaeobacteria. I haven't looked at this data yet because it's my colleagues that are working on it.
Okay.
I don't remember that we saw this high in the Tanzanian and Botswana samples that I would show you. Otherwise, I should have highlighted that. But I think it's interesting to know that you find this, and we will look at whether we see this in the Cameroonian samples. At this moment, I think we didn't find that. We didn't find this observation in Tanzania or the Botswana samples now.
Interesting.
Thank you so much. But interesting to highlight this.
For sure.
Yeah.
Other questions?
I do have one more question actually for you, Eric.
Yes, please.
When you guys are moving around subsistence, or some of the diets at some of the populations that we have are pretty much on a strictly cassava only diet. Are any of your populations, are they more diverse in diet, or do you have some groups that are, you know, pretty much only cassava?
Looking at Cameroon, for the populations that we have, basically, I would say that's why it was a little bit difficult for us to streamline to a particular diet. They're a little bit more diverse for the population.
Exactly.
Far, yeah, even from Botswana or Tanzanian population, and more even from Cameroon. You see that there's a little bit more diversity. So I would say cassava, yes, cassava is really in the forest part of Cameroon makes most of that diet, but then you also have plantains, you also have other foodstuffs. So yes, not really. We didn't find a population that you can really say this is that. I think that's one of the difficulty in the population that we have.
Yeah, that was So luckily we've stumbled across a particular region that relies, I'd say, nearly exclusively on this. So I mean, that may be affecting some of the data that we're seeing as well. I mean, and they're consuming bitter cassava, so it's really high in cyanogenic compounds, much higher than say sweet cassava.
Right.
So anyway, it'll be interesting to see what that looks like. based on
Exactly.
diet alone.
Yeah.
I have a question.
So Eric, oh, go ahead.
Hi, this is Anthony Moira from UCSF. Eric, great presentation. Thank you. It's interesting the similarity between Botswana and the U.S. population, and you mentioned a little bit about the possible economics of that. Any thoughts about how urban populations, say urban Tanzanians, urban Cameroonian, and people who are eating more, sort of living more Western living standards, how that would look like compared to U.S. populations?
Thank you so much, Anthony, for this question. And that is a little bit downside in a study or something that we are looking into also. We look at rural population here, and we are thinking about collecting samples in the urban areas so that we can see, especially as you highlighted, you indicated with this observation from Botswana, we don't have samples from urban areas, but we are planning to compare, to look at urban areas, and maybe from urban areas we will find the observation that they will be more closer to US samples when you look at urban Tanzanians. So yes, that's something we have also, we've thought about it and we plan to do.
Greg, you mentioned the low BMI, and I was just wondering in the future for extending the studies, and do you think you're going to be able to make some correlations with some health outcomes like obesity and diabetes in some of these populations?
Good question, Kim. So if I come back to look like at Cameroon and something that I'm particularly interested in, the Fulani population, the Fulani are pastoralist population that we have studied from Cameroon. And this Fulani have been shown in other populations in Burkina Faso and in Mali that they seem to have a more boosted immune response to malaria. So they seem to be more protected against severe malaria for reasons that are not completely known. But when they look at the they seem to have a more inflammatory response or some responses, specific responses sometimes to malaria. And surprisingly, it's also been shown that there seem to be a higher prevalence of diabetes in this population. And we have a cohort, a small number of diabetic patients. But we plan that when we go back to the field, we are going to recruit more diabetic patients from this population, and then we will be able now to look at some of this analysis and the gut microbiome also related to diabetes. So yes, we are looking at that, and we know that gut microbiome has been associated with obesity also. But this is a population that you have a little bit higher incidence of diabetes in that population, but the population compared with the Bantu neighbors, they are less on the obesity side. In fact, obesity is very rare in this population. Yes, so those are aspects that we plan to look at it in our analysis scheme.
Thanks.
Hi. Hello. Can you hear me?
Yes, please.
Yeah, go ahead.
Okay. I have one question. Thank you for the good presentation.
Thank
when
you.
you started the talk, one of the main aims was to see if the observed differences were related to diet, geography, and or ancestry.
Yes.
So at the end of this study, what can we attribute, how much of the variation is attributed to diet, geography, and ancestry? Because these three factors are not necessarily independent, because the results show that the US is different from Tanzania or Botswana, but all these locations give you information overlapping about these three factors. Diet will be different, geography will be different, ancestry will be different. So they are not necessarily independent factors. How do you attribute, how much of the variability is due to diet, due to geography, due to ancestry, and so on.
Thank you so much. Great question, and that is a challenge also for us. When we started the study, we were thinking that what we were hoping for was that with the different subsistence, we thought diet varies between these different subsistence groups, and that what we were going to see was that the contribution of diet would be more than the contribution of the other factors. But surprisingly, that didn't happen. We look at instead, geography was showing up as affecting gut microbiome. And like you said, when you talk about geography, it's basically a combination again of diet and relatedness. And I think this is this is a challenge to really distinguish what is the contribution of which part, or whether it's diet or stress, because we didn't even collect specific dietary information, which we want to get a little bit closer to this. And that's why shotgun metagenomics will also have an added advantage when we look at this, which is going to tell us a little bit more about other exposures. But like you say, it's still not very clear at this point what is the contribution of ancestry, what is the contribution, because all of that still comes to geography. Great observation, thank you.
Okay, I guess we will look this further in the future, hopefully, in the next presentation. Thank you for the answer.
There was one question in the chat box there. Says North African populations are highly mixed and highly influenced by the Western diets, local habits, other things. Do we expect to find similar results if we undergo a same study?
Can you say that again, please?
Yeah, so it's a question in the chat box. It's saying Northern African populations are highly mixed and highly influenced by the Western Arab and local diet habits, high automedication, high prevalence of high blood pressure and diabetes. And do we expect to find these similar results if we undergo a same study?
Thank you. I think what will speculate, like you highlight already, the differences between what you obtain in North Africa and compared with sub-Saharan Africa or the southern part of Africa. I would expect the northern part of Africa maybe closer to what you have we observe in the US, but that would be an interesting thing to look at, because then at that moment you look at the contribution of relatedness also in microbiome composition, of genetic ancestry in microbiome composition. It will be good. It will be interesting to look at that, because their genetic ancestry is also a little bit different from Southern Africa, from south part of Africa, and from the US. But generally, I would expect from the other contribution less pathogen exposure. Like you said, the disease is different, and yeah, I expect differences. But that's something that really it would be interesting to bring a cohort from that and look at the picture. One thing I think is there's still a lot that is not known about microbiome, and sometimes, like we were surprised here with subsistence not showing up, you might be able to see some great. We might have other surprises.
And Eric, you mentioned that the geography seemed to be, really key, and I guess the geography could be a gross reflection of lots of different types of exposures, kind of all combined. I don't know how in the future how much you might be able to tease those particular factors out further.
Thank you, great question. So one thing, like I said, we are looking into it is pathogen screening. Is another aspect of it to look at whether there are exposures to particular pathogens in particular areas compared to other areas. Another thing we are looking onto is also collecting, like I said, we didn't collect dietary information, So we're looking more into what specific dietary information. And when you do this longitudinally, combining, we look at only one season, because sometimes there are also sometimes gut microbiome differs between seasons. But when you take a longitudinal study, also crossing over from one season to the other, you might be able to look at some of these aspects, climate and everything that comes into geography also. And like I said, yes, shotgun metagenomics is the way to go, and this will tell us a little bit more about some of the exposures, the differences that can account for these differences. So that's the way we are looking at it, and how the approach is going.
Okay, great. Any other questions? Okay, well, if not, thank you so much, Eric. That was really great. And good luck in the future with teasing all this out.
My pleasure. Thank you for having me.
Thanks,
Okay.
Eric, and thanks, Kim.
All right.
Thank you.
Thank you, everybody.
All right. Okay. Bye.
Bye-bye.
Bye.